It's an honor for me and a great pleasure to introduce Thejasvi Venkatachalam, who's a Rutgers class of 2022, a major in the molecular biology and biochemistry program here at Rutgers. We had the privilege of hosting Thejasvi as a summer research intern in the RCSB Protein Data Bank this year, and she did a spectacular job and played a critical role in helping to prepare for the IQB summer boot camp on the coronavirus, during which we trained more than 30 undergraduates in bioinformatics and analysis, three-dimensional analysis of protein structures. And then she contributed to the summer research program that you've heard about, looking at evolution. So her topic is going to be the only validated drug target for SARS-CoV-2, the RNA-dependent RNA Polymerase. Thank you, Thejasvi.
Thank you, Dr. Burley. So hi, everyone. My name is Thejasvi, and I'm really pleased to be speaking to you all today. So the subject of this talk is the RNA-dependent RNA Polymerase, or the RDRP for short, as I'm going to be calling it throughout the talk. Okay. So similar to the previous talks, I'm sure, in this talk, we will start by discussing how the RDRP works and its role in the virus life cycle. Then we'll talk a little bit about the mechanisms of action and the enzymology of the RDRP and what we know about the structure of this protein complex. Following this, I'll talk a little bit about the unpublished data about the RDRP and how it has evolved during the pandemic and what lessons we can learn from this. All right, so the RDRP is composed as a heterotetramer. It's made up of an NSP7 protomer, two protomers of NSP8, and the catalytic subunit NSP12. So I'm sure you've already seen this diagram in the previous talks. The non-structural proteins NSP7 and 8 are encoded in open reading frame 1A, and the RDRP, or NSP12, is encoded in the open reading frame 1B. So as the name implies, this is the complex that's responsible for copying the viral RNA genome, and it also copies the 9 canonical subgenomic RNAs. So as you can imagine, that makes it a very important subject to study. All right, so as I mentioned just now, the RDRP is a heterotetramer, and it's made up of NSP7, 8, and 12, which are 83 and 198 and 932 residues, all respectively. So NSP12 has an active site that binds one turn of the double-stranded RNA. So if we look at the structure here, this is just of the structure 6YYT. The light blue is NSP12. These two arms here in the darker purple are NSP8, and this lighter purple here is NSP7. And then in the back, we have a double-stranded RNA primer. So NSP12, as I mentioned, has the active site. And in the NSP8 protomers, as you can see here, they flank the RNA and they prevent premature dissociation of the RNA from the RDRP complex. And this is a very important function for effective replication to occur. So here's a ribbon diagram of the RDRP bound to the RNA. So same thing again, NSP12, the two arms of NSP8, NSP7, and the RNA. And I also pointed out where remdesivir would be binding, but there is a structure of that will be coming up. So NSP12 has 3 main domains: the RDRP-associated nucleotidal transferase domain, the interface domain, and a C-terminal RDRP domain. So the NSP12 active site is observed from residues threonine 611 to methionine 626. Additionally, we see that aspartic acid 760 and 761, they're important because they recognize the 3' end of the RNA, and without this, we can't have effective RNA synthesis. Additionally, we also see that in the alpha helical arms of NSP8, There are a bunch of positively charged residues, lysine and arginine, as you can see here, and these all interact with the RNA as well. And from these, it's been found that lysine 58 is really essential to RNA synthesis, and without it, the function of the RDRP sort of goes away. Okay, so these two figures are just showing what I was just talking about. So here on the left, we have NSP12 shown in yellow, and the active site I labeled in green just to point it out, and all the residues are shown in ball and stick format. And then over here on the right, we have the two arms of NSP8 in pink and orange, and all of the residues that are involved in interacting with the RNA are shown in ball and stick. And the labeled residues are actually the ones that we observed mutations in during our analysis, but I will get to that in a little bit in the talk. So several structures have been released up until this point that contain the RDRP. And the first one of these was 6M71, and this was released April 1st of this year. So 15 structures, to be precise, have been released up until now. And of those, 3 of the structures have bound drugs. So the first one is one we've all heard about. It's remdesivir. It's the approved drug for SARS-CoV-2. And then we have favipiravir, is an influenza drug. And then finally, we have suramin, which is an anti-trypanosome drug, which treats African sleeping sickness. From these, we know that remdesivir has been an approved treatment for SARS-CoV-2 from the US FDA emergency authorization use. And this makes RDRP a validated drug target. So on the right, we can see here a close-up of remdesivir bound to the active site of NSP12. And through the analysis of the structure, we can see that the listed residues here are the ones that interact with remdesivir. From this, we can see that the bolded residues here, aspartic acid 623, that is involved in the NSP12 active site. And then the two 760 and 761, those are the ones that I mentioned that bind the 3' end of the RNA. So this kind of gives us a little bit of an idea of how remdesivir acts and how it interferes with the action of the RDRP. We also know that remdesivir is not really the best drug to treat SARS-CoV-2. It does work a little bit, but it's not the best since it was originally used for Ebola and SARS-CoV. But this structure is a really good starting point for us to look into the discovery of second-generation RDRP inhibitors and how there could be potential for any drug resistance that may arise in the presence of a more potent inhibitor. So now that we've established this information, we can start to talk about how this complex evolved during the pandemic. So I'm sure you've already heard a little bit about how the project was conducted in the previous talks. So I will gloss over this a little bit. I will mention that the RDRP structural modeling and energy calculations, for this, the APO structure 6YYT was used. So that was the structure that we were looking at until now with the NSP12, NSP7, 2 copies of NSP8, and the RNA primer. Okay. So for NSP7, we looked at 48,670 viral proteomes, and of these, 98% had NSP7 unchanged, and there were only 68 unique sequence variants, of which most were single mutant variants. And then when we look at the modeling and the energetics, we saw that 25% of the mutations were found in the core, 53% were found in the boundary region, and 25% were found in the surface area. So we see that it's most marginally destabilizing. And then for NSP8, we had both of the arms were included in the energy calculations, but when we looked at it, it was just taken into account as the actual transcript of the NSP8. So we had 48,335 viral proteomes, and of these also mostly were unchanged. 99% had NSP8 unchanged. We saw 144 unique sequence variants, 140 single mutants, 2 doubles, 1 triple, and 1 quintuple. And then from this, we saw that 5% of the mutations were found in the core, 37% were found at the boundary regions, and 63% were found at the surface. Now moving on to NSP12, this is the catalytic subunit, which makes it the most interesting to look at. In this one, we saw 44,203 viral proteomes. And of these, only 23% had NSP12 unchanged. And we're going to see why in a little bit. So from these, we had 249 single mutant variants, 424 doubles, 51 triples, 3 quadruples, 3 multipoint. More than 97% of these unique sequence variants had the mutation proline 323 to leucine. So this represents sort of a new NSP12 clade that emerged during the course of the pandemic. I think it was somewhere earlier on. And when we look at the doubles and the triples and all of the multipoint mutants, most of them have this mutation in it as well. So for the modeling, we saw that 9% of the mutations were found in the core, 63% were found at the boundary region, and 29% were at the surface. All right, to summarize this a little bit, when we looked at NSP12, we tried to see if there were any mutations to any of those important residues that we talked about. And fortunately, I guess, we found that there were no mutations that affected the remdesivir binding residues. So that's sort of encouraging news. And then for the mutations that affect the active site, we saw these changes to histidine 613, leucine 614, methionine 615, tryptophan 617, tyrosine 619, and alanine 625. So these were the ones that were changed, but we didn't see anything for remdesivir binding residues. For NSP8, we saw, like that picture that I showed earlier, we saw that these were the residues that were changed, lysines 37, 40, and 61, as well as arginine 51 and 57. Most notably, we didn't see any mutations to that lysine 58 that I mentioned was very essential to RNA synthesis. So that sort of reinforces the fact that that is required. For NSP7, we didn't see, well, we saw some mutations that affected the protein-protein interfaces between NSP12 and NSP8, but there wasn't really anything notable, nothing changed. There was nothing that stood out or that stayed the same throughout all of the unique sequence variants that we looked at. All right. So at this point, it would be apt to discuss how the RDRP works with other proteins to replicate RNA. So one of these proteins would be non-structural protein 13, which is the RNA helicase. So much of the RNA genome is actually double-stranded, or it has a secondary structure, so it needs to be unwound before the RDRP can go on to copy it. So shown here below is the secondary structure of the 5' untranslated region of the SARS-CoV-2 genome. This was just taken from this paper here. And so we can see here, this is where the start codon for NSP1 actually is. So there's a lot of secondary structure to unwind before the RDRP can start copying any of the genome. So it comes in and it unwinds the viral RNA, and then this allows the RDRP to actually come in and copy it. So to the right here, this is a structure that shows this whole complex together. So this is the PDB ID 6XEZ. So this is pretty much the same as the other structure that we were looking at. It has the NSP12, 2 arms of NSP8. In the back there, in the purple, we see NSP7. There's the RNA. And then these 2 slightly different colored units are NSP13. So this is where it would unwind the RNA and then allow the RDRP to actually copy the genome. And it's also worth noting that NSP14, which is the proofreading exoribonuclease, is probably also part of this complex. So that would also be interesting to look at. So all in all, some key lessons that we've learned here, this proline 323 to leucine substitution in NSP12 is really a distinct evolutionary clade that emerged, and it's worth looking at a lot. And then in terms of NSP7, NSP8, NSP12, we didn't see any substitutions that were really that common, except for this one. And that's probably largely due to the proofreading activity of NSP14. And to sort of go along with that, we didn't see a lot of, we saw a lot of preservation of the active site and the protein-protein interface, just because it is required for the virus to be able to survive. For the structure of remdesivir bound to the NSP12, we see that this is a really important structure for us to study, because it gives us an idea of how new drugs can be modeled to actually inhibit the RDRP more effectively, and and this can also provide clues for potential sources of drug resistance that would come up as a result of this. So I would like to extend my acknowledgements to the Rutgers PDB. My special thanks to the PDB director, Dr. Burley, as well as Dr. Kare and Dr. Datta. They were really great mentors and they helped lead the project, and I owe a lot to them. And then I would also like to thank my teammates, Luz Alvarado and Helen Zhang. They really contributed a lot in this analysis of the RDRP, as well as other structures that we looked at during the summer. I also want to thank Stephen Arnold, who I worked with a lot during the summer, and the entire boot camp, all of the participants and all of the mentors. So I'd like to open up to questions now.
Thank you very much, Thejasvi. So let's see, you have a question in the chat from Jeffrey Yang, who says, because remdesivir binds to SARS-CoV-2, but was originally developed against Ebola, does this suggest that the polymerase of SARS-CoV-2 and Ebola are similar? Are Ebola and SARS-CoV-2 potentially related to one another? Does this provide a rationale for broad spectrum antiviral agents against polymerases, or was this just a coincidence?
Well, I know that it was, it applies to SARS-CoV as well. And so I think that's why it was really applied to SARS-CoV-2. I'm not sure if Ebola, if the polymerases of Ebola and SARS-CoV-2 are similar. And I mean, I guess if it was, that would be, evidence to the fact that it might be a good antiviral against all polymerases, but I'm not sure if that is actually the case. I'm not sure if anyone else knows.
Would anyone like to pitch in? I don't know, Eddie, are you still there? Maybe, I'm sure Eddie will know. Or anyone else?
This is Sanjay.
Yeah.
Yeah. So what I would like to suggest, like remdesivir is basically having a product architecture which has a triphosphate. So in case of any polyproteins or proteins where we have this polymerase activity, because mainly it is preventing this polymerization, the incoming nucleotides will be prevented because of this structural similarity. So that is why not only in Ebola virus, in most of the other viruses where this polymerase activity will be there, it can be prevented using this remdesivir drug. But this is why it is also creating a problem, like why it is not covering most of the patients' prevention of COVID-19, because it is not that much specific. Or for this particular SARS-CoV-2, we should design an optimized remdesivir structure so that it can have more potency, like in nanomolar or something, so that the affinity is increased.
So it may provide a good starting point for developing RDRP inhibitors for RNA viruses, but it's a weak inhibitor and not as effective unless it is made, unless it's kind of specialized towards the target. Yeah, that makes sense. Okay, next question is from Sanjay. Says NSP12 also has a NERA end domain. Can it be a site for drug target? Could you find mutations in that site as well? Could you find mutations in the zinc finger as well?
Yeah, I mean, that would make sense that this would be a good site for a drug target. I can't remember right now if there were mutations in either of those regions. It's been a while since I looked at the actual data, but yeah, I'm sure that there is probably something. But I want to note that like a lot of these unique sequence variants that we saw were only observed like once or twice, especially if we saw them in like important regions. So that is worth looking at.
Speaking of how many were found, Martha Soto asks, was the proline to leucine mutation found in all geographical versions of the virus?
I think from what I remember, I think it originated in the United Kingdom, and then it actually spread to a lot of different places. So it was observed in a lot of geographic versions of the virus.
Okay. And Javi asks, NSP7, NSP8 is thought to act independently to NSP12 as a primase. It could be that K58 mutation is knocking out this activity, not the RDRP. Yeah, that's an interesting thought. So
Yeah.